I want you to imagine that you're holding a plastic bag full of water. There are holes in this bag and the water is leaking out. But you have one band-aid, so you place it over one hole and now you've stopped that leak. However, there are still other holes in the bag and the water is rapidly flowing out, and you know that the only way to stop it completely is by covering every hole. Now I want you to imagine that that plastic bag full of water is a tumour, and each hole is a different way in which cancer cells can grow and survive. And that one band-aid that you used is a standard cancer therapy. Currently, this is what we are doing in medicine, only covering one, maybe two holes, leaving the others open and giving cancer a chance of survival. In order for us to cure cancer, we need to cover every hole possible, and we can only do that by understanding every way in which cancers grow. Tumours grow in nutrient-poor conditions, and they need to be really sneaky in order to survive. And one thing that they do is they trick the cells around them into helping them out by sending what we call exosomes. Exosomes are small packages released by every cell in our body, and they carry messages from one cell to another. Under healthy conditions, this is a great way for our cells to communicate. But in cancer, tumours manipulate this system to send bad messages to healthy cells. These bad messages can force healthy cells to become cancerous, feed the tumour, or even help it evade the immune system. As you can imagine, this is a huge problem. And even though we know exosomes are helping cancer, the bigger problem is that no one understands what is controlling the release of exosomes from cells. This is the aim of my PhD project, to try and identify what protein inside the cell is controlling exosome release. And I have an idea as to which protein it might be. And one of the ways that I've tested my theory is by blocking this protein in cancer cells and simply counting the number of exosomes that are released. And what I found is that when I block this protein, there is a huge increase in exosome number, indicating that this protein is highly likely to be regulating exosome release. Additionally, I'm also on my way to showing that we can alter the messages inside the exosomes by also targeting this protein, giving us the ability to turn exosomes carrying bad messages into exosomes carrying good messages that can kill the tumour instead of helping it. But why is this so important? Well, if we can understand what is controlling exosomes in the cell, we can target it and either block exosome release or alter the messages carried inside, ultimately starving the tumour, stopping it from spreading to other organs, and giving patients a better chance of survival. Every advancement that we make in cancer research, every new target that we identify is a new band-aid that we can make to cover another hole in that plastic bag full of water, taking us one step closer to the ultimate goal, curing cancer.